Thank you. And thank you all for coming tonight. I hope something, somewhere in this experience, you'll see something that sparks something that made it all worthwhile. Whew. And, it's natural to wonder, if you're switched on, whether I'm an expert, whether I'm worthy of presenting this material to you, and what my motivation is for doing this, for being here. And of course I'll oblige because it's important for our story tonight. I'm part of a group of people who have dedicated our lives to understanding, uncovering, and sharing the story of human origins and humanity's place in nature. So that one tiny, I'm going to use this laser pointer like crazy because it's so fun, that one teeny tiny twig on the tree of life that ended up like this. So I'm a biological anthropologist, I'm an evolutionary anthropologist, and it's thanks to the Leakey Foundation that I can say that. Here you see me excavating with my friend Will. I'm excavating the best fossil I have ever discovered. That's what it looked like. Well, it was a little dirtier than that when I first saw it in the rock outcrop. And there were a few more bones sticking out like this at me that had crumbled away, and then I bagged carefully and marked carefully so that I could glue them back together. But when I brushed at it, this is what I saw, and I hope you can tell that those were ribs, and that this is a vertebral column. It's part of the spine, the bones of the spine. And because the way that the scapula, or the shoulder blade, was oriented, we thought, well, if we dig over here, maybe we'll find the head. So we dug over there and found the head. I hope that was obvious. And here it's a sagittal view of the head, of the cranium in the rock. Here's the top of the skull. Here's the snout, the tip of the nose, the teeth and the maxilla. And you can barely see the orbit, the hole where the eyeball was. And when it's all cleaned up, this is what it looks like. It's a really nice partial skeleton with a really nice cranium. We thought maybe we would find the mandible when we looked down the gully because it might have fallen out of the rock, and we couldn't find it. And we thought maybe, decades earlier somebody had collected it and put it in the museum and it would fit on nicely, and we didn't find anything like that. So unfortunately, this is all we got, but really, this is really nice. But I hope you can tell that, although I'm an anthropologist, the best fossil I ever discovered is not a human. And I hope you can tell that the best fossil I ever discovered is not a primate. Any guesses? And let me give you an age, 20 million years old. You're not going to guess. I didn't know what it was either, so I'm not going to make you guess. It's an ancestor of this guy. Now, does anybody know what this is? Yeah, no you don't. How do you know it's a hyrax? Because Beth says they're awesome. Okay, what tips you off that it's not a rodent? Thank you. Or a lagomorph, a rabbit or a hare. Yes. And so did you know, okay, you knew it's a hyrax, but did you know that this is where it fits on the mammalian phylogeny? It's more closely related to elephants and other things than it is to rodents. So we're actually more closely related to rodents, or Beyonce is. So I'm just trying to suck you into the joys of paleoanthropology. And that was a little story from Razinga Island, Kenya, where I've been doing paleontological fieldwork for the last 6 years or so with generous support from the Leakey Foundation. And our sites on Razinga Island are right here in Lake Victoria. It's this funny little shaped little island in this beautiful lake in the beautiful region of western Kenya. And that star is on our campsite, but there are fossiliferous sites all over the island. And this island has been famous for a very long time, thanks in main part to all the work that Lewis and Mary Leakey did on the island in the early part of the 20th century. And the middle there is a very nice photo of what we call Mary's skull, the famous proconsul Hesleni skull that is an early ape from about 20 million years ago. You can hold it in the palm of your hand. It's a very small-bodied, very monkey-like ape that is probably related to, somehow, the earliest apes in the ape family tree. And it's so famous in Kenya that it made a postage stamp. So we've been, oh, and I want to put proconsul in evolutionary context for you. It's here about 20 million years ago, and notice how nicely that sits on the phylogeny, the hypothesized phylogeny for the origin of apes here on the primate family tree. And here's a nice, kind of old right now, but still really good reconstruction of what proconsul would have looked like walking on a tree branch. So if you're wondering where the name comes from, when Lewis and Mary Leakey named proconsul, it was a hypothesis from the start that it was an early ape, that it was a proconsul. Consul was, for a long time, kind of like jumbo for elephants, was a common name for entertaining chimpanzees, like these cute little guys. And since then, since we've been working at these sites, we have found a lot more primate specimens. There isn't just proconsul on Rosinga Island. There are many more, not many more, but several other funny little fossil ape taxa, or fossil, what we call catarrhines. We're not really sure how they fit in with the evolution of apes. But we found some nice ones, mostly jaws and teeth, but we've also have this little arm here. Here's a little bit of your humerus, your distal humerus, the elbow, and a radius and ulna. And of course, the preservation on this island is fantastic. This is just one of the many leaves that are preserved on Rosinga Island, I hope you can see here. And we're lucky enough to have a huge collaborative team that includes someone who is an expert at studying the shape of leaves, that you can actually estimate temperature and rainfall based on the shapes of leaves. And he's done that, and we have a nice estimate for at least where these fossil leaves are preserved of a really hot, really wet rainforest that's linked to the time that these early apes were living, and that makes a lot of sense. Apes like rainforests. But we're hoping to find some more of these fossils like these crickets and like this chameleon. See how great the preservation is? This is just the smattering of the amazing kind of fossils we can find on Rosinga. And so this is why we hope to be able to reconstruct the paleo environment in really, really high resolution detail, to get a really true snapshot of this part of our evolutionary history, albeit an early part of our evolutionary history, but a really significant part of it, the earliest apes. And what got me studying, what kickstarted this work, this field work on Rosinga Island, really was my dissertation research, also supported by the Leakey Foundation, which was studying this amazing collection of partial skeletons that my doctoral advisor and his colleagues had excavated in the mid-1980s. And it had largely gone unstudied. It was kind of sort of hibernating in the museum for a while, I think because right after they excavated these, they found the Nereocotomy Turkana Homo erectus boy. And when you find a Homo erectus skeleton, even if it's just one, 10 partial proconsul skeletons might get put in a drawer for 20 years. And so I was fortunate to do some original research on this. And because this collection represents proconsul infants to adults, I thought it would be really fun to try to figure out if proconsul, this early ape, was growing up like an ape, like extant apes grow up, with the same patterning and scheduling and timing, which is different from how monkeys grow up. And I wanted to see if it grew up like a monkey or an ape. And I'm still working on that. And while I've been working on that, I couldn't help but wonder, because I'm an anthropologist, what this has to do with how humans grow up. And I couldn't help but ask the age-old question, why are babies born so helpless? And so that got me onto another research project that's a collaboration with some other colleagues. I can't not show this. I love showing this Damien Hurst sculpture about human pregnancy. So the classic explanation for why babies are born so helpless has been for decades that the human birth canal is so narrow that the baby is born early to escape in time. And that's a really pared down version of the explanation, but we've got to get to genomes. So, but that's basically that it's a pelvic reason, that it's because we walk upright. And instead, my colleagues and I looked at a lot of different kinds of data, a lot of current evidence since this hypothesis had been first offered in the middle of the last century. And it doesn't look so much anymore like it's a pelvic constraint that makes babies born early. It looks like babies are born when babies should be born for apes, but that humans have an energetic or a metabolic constraint, and it's really all that mother can do. And when the fetus is taking more than mother can give, the fetus is born. And it's no longer a fetus, it's a baby. And so we called this hypothesis very cutely the egg hypothesis for energetics, gestation, and growth. And we're very proud to have had this published recently in PNAS. So, now, look at the title of my talk. What is someone like me doing, standing here talking to you about genomes and how yours is showing and how human origins just got personal? Okay, I live and breathe evolution, and not just because I'm one of its products, but because I study it and I teach it. And if I'm really going to understand what fossils and metabolic constraints have to do with evolution, I've got to understand genetics. We all do. We've got to understand the biological information that's fundamental to our existence, to our lineage's endurance, to our humbling stint in this unbroken thread of life. So anyone who wants to know anything worth knowing has got to know a bit about genetics. And as much as possible, tonight, we're going to use art instead of pictures of molecules and infographics. And since I really like album cover art, voila, I hope you recognize this one from the Rolling Stones. There will be some weird ones, I'm warning you. I thought it was appropriate given the nightlife atmosphere to be a little rock and roll. So instead of the Big Bang, let's start with the Big Banana. Split from human ancestors about 1.4 billion years ago, sharing about half of its genome with us. Of course, that doesn't mean it's half human or that humans are half banana. It just means that if you put our genomes side by side and compare all the C's, T's, G's, and A's, the bases that are the letters that spell the code, and you focus on the differences, well, you can estimate how long it took for those differences to accumulate. And assuming, quite fairly assuming, that it's a clock-like rate of mutation, or of change, then you get that at about 1.4 billion years ago, those differences began accumulating between banana ancestors and human ancestors. So, between our ancestors and dogs' ancestors, those differences began accumulating around 95 million years ago. And we've been evolving separately ever since. And for monkeys, we've been separated since 43 million years ago. And of course, by around 6 million years ago, chimps and bonobos had just about enough of it. Thank you very much. And started their own evolutionary journey apart from ours. And since then, we've been walking and running upright. And we've evolved these ridiculously large brains, which enabled this. That's thanks to my husband. Yes. The molecules of inheritance, DNA, many of which that we share with bananas, dogs, chimps, and are also the instructions for how our bodies grow and how they function, and maybe even how they die. Much of the differences between me and a dog or you and a chimp boil down not to differences in proteins or functions, but in the regulation of those proteins and functions, the timing, the duration, the amount, the locations, the combinations. Since we can assume that identical twins have identical genomes at birth, it's clear that DNA is the blueprint for the body. And when we see differences between bodies, something you don't see much of between identical twins, it's a good hypothesis that those differences are due to DNA differences, to being conceived at a different time from the same parents, or to coming from different parents. And to get down to the details of those differences, to what makes us each genetically unique, to see what that's all about, we now have enterprises like 23andMe. And with the products and services offered by companies like this, it's never been easier. I sound like a commercial. I really don't want to. But it's never been easier to see thousands of regions of your own genome. On your sofa, in your jammies, with a quick cyber swipe of your credit card, you can have a 23andMe spit kit sent right to your door. And I want to show you some of what it's all about, in case you don't know. Many of you might have already gone through with this. I would love to talk about it with you afterwards. But I want to show you some of what it's all about, but I also want to demonstrate why, as a teacher, I offer the opportunity voluntarily to my students in my human origins and my human variation courses at the University of Rhode Island. So if you watch nerdy PBS programming, then you're already familiar with 23andMe, at least a little bit. Skip Gates's recent series, Finding Your Roots, featured results from 23andMe, which gave us, for example, this whole new way of thinking about the 6 degrees of Kevin Bacon, who was one of the participants in the genome analysis on the show. And this diverse family, this is a very diverse family depicted here, beautifully demonstrates how we're all connected a lot more intimately than we might think, than our exteriors and than our behaviors might lead us to believe. And here's how it works. You sign up on the website and a spit kit arrives. You follow the easy steps, spitting into a tube, and then send it back to the self-addressed prepaid, with the self-addressed prepaid packaging to 23andMe headquarters, which is right down the street from you guys. And a quick review of some familiar terms and maybe some newer, unfamiliar ones, but I hope they make sense. What 23andMe counts on is that most of your cells contain, in your saliva, contain your full genome. And they do, mostly. And in the cell nucleus, they've got your nuclear genome or your nuclear DNA, and it hangs out in a goopy molecular mess most of the time. But right before cell division, which is part of normal growth and body maintenance, and it's happening all over your body right now, right before the cell divides, the DNA bunches up into nice little neat chromosomes like this, and you can see them. And I like to think of this view, this view of your genome and its nice, clean chromosomal glory, sort of like Dolly Parton. You can only see her when she's all dolled up, literally, and that's true for your chromosomes too. So you have these 23 pairs of chromosomes, one from your mom and one from your dad, one from your mom's egg and one from your dad's sperm. This is when everybody squirms. Too much already. That's the last time I'll say your dad's sperm. And each one from your mom is a mosaic of pieces. Okay, this is where it gets deep and heavy. Each chromosome from your mother is a mosaic of the pieces, of the chromosomes of her mother and her father. And each chromosome from your father is a mosaic of the pieces of his mother and his father. And that, along with a couple hundred, which is like nothing for the 3 billion base pairs, but that along with a couple hundred brand new or de novo mutations that each and every one of us inherits is why each of our genomes is unique. So these chromosome pairs are homologs, meaning, bear with me, we're going to get to Snips in a second, and that's important. Just bear with me. I want you all to be here. These chromosome pairs are homologs, meaning they contain the same genes and other bits structurally, but they do not contain identical codes. That would mean your parents married each other's clone, and that would be, or not, or made you, let's not think about it. For example, you have the gene for earwax, something we definitely should think about, earwax. You have the gene for earwax here on the 16th chromosome. And you've got one version from your mom, and you have a version of that same gene from your dad. You have 2 alleles for every gene and every other stretch of DNA in your genome. You could have identical alleles from each parent, or they could be different. It all boils down to how the A's, C's, T's, and G's line up. And that's true for every part of your genome. 2 copies, one from mom, one from dad, and that's what's known as your genotype. Your genotype. And sometimes those 2 copies work together. Sometimes one works alone or dominates, or is dominant, I should say, and the other isn't expressed. Depending on the behaviors of those alleles and how they do or do not behave in different contexts, including your external environment, those alleles, your genotype, determines a lot or a little of your phenotype. You only have those 2 copies, but for many stretches of the human genome, more than 2 copies exist out there in the world. Still, each of us can only carry 2 of the possible variants that exist in the whole species. When alleles vary by only 1 letter, they're called Snips. SNP is the acronym. Single nucleotide polymorphism. Snips. So the 2 known alleles for earwax are Snips. There's waxy and there's crumbly. Yes. And they differ by only 1 base pair, by 1 letter. They are Snips. And a lot of human variation boils down to Snips, or is affected in large part by Snips, by variations that are only at 1 base pair. Now let's stop looking at this one and get down to business. So your spit is scanned in, and then your DNA is extracted from it, and then your DNA is explored using a SNP chip, which is basically a bunch of SNPs that are known already. And if your SNP sticks to it, you have that SNP. It's based on the complementary nature of the DNA molecule. And if your SNP sticks, you got that SNP. If nothing sticks to that SNP on the chip, you don't have that SNP. That's as easy as that. So it's not sequencing any of your genes, 23andMe. It's genotyping you, or your spit. It's genotyping your spit. All right, then depending on what you decide or what you signed up for, your DNA is stored at 23andMe in their laboratory, and then finally your results are generated from these machines and are reported on the website. They're not sent to you in the mail. You have to go to the interface on the internet and you look at your report on your computer. And what's in your report? Yes, your earwax snips. Whether you have the alleles for curly or straight hair, what eye color alleles you have, whether you have the SNP for tasting some bitter compounds that other people cannot taste, your alleles concerned with when you wake up in the morning, your alleles for lactase persistence, whether you can digest lactose worry-free as an adult, whether you've got the alleles for fast twitch muscle fibers, making you a sprinter, cardiovascular disease risk based on genes, and many other genes that are simple or complex, meaning that they are affected by multiple SNPs and or environment as well. So things like diabetes risk, obesity, a host of cancers like melanoma, and locked away to be unlocked if you choose is your risk for Parkinson's and your risk for Alzheimer's. And in a separate report, you're told of your risks for negative or positive responses to different drugs or medical treatments. I Googled most psychedelic rock albums to get this one. And then, of course, and we're not talking about that, though. We're talking about medical drugs. I mean, upright citizens type stuff. And then, of course, there's a whole report on the ancestry history of your SNPs, linking them to people around the world and inferring common recent ancestry with them. Now, does it work? And I'm sorry that you cannot see this in the back. I did not expect such a big room. But that just means we won't spend much time on it, and that's fine, because I do not think we need to talk about me for very long in this experience here. But I want to show you my report, a little bit of my report, so you can see why I'm talking about it today. And the first thing I want to do is, this is supposed to be animated, and it's not supposed to show you my result yet. We're supposed to sit here and guess, so I'm going to go backwards. Pretend you didn't just sit there reading that. That was supposed to come up. Let's guess my genotypes based on my phenotypes. So let's guess my earwax. Well, I'll just tell you it's waxy. And my hair is curly. My eye color is blue. I do taste bitter, and I am lactose tolerant, and I'm sort of a sprinter. Is that what 23andMe found? Yes. And more. I'm not going to talk about all of these different reports here, but that's definitely what they found. There are no surprises, which is great, because that means they are mining the right Snips to explain my phenotype. And it means that, yes, literally, my genome is showing and is impossible to hide it. Now, let's look at my blood type report to see if they got that right, because I actually know my genotype for my blood type. They report on the ABO blood type and the rhesus system as well. And I know that my blood type is A positive. I know that my genotype is AO because my mother is O. And so that means she's OO. So I had to get an O from my mom, and I know I'm type A, so I'm AO. Look at that. They got it right. And I'm A positive. I'm positive about that. They got that right. So here's what I know about diseases in my family. We've got glaucoma, macular degeneration, emphysema, strokes, heart attacks, ovarian cysts, skin cancers, colon cancer, psoriasis, breast cancer. And if we look at my elevated disease risk based on my gene-based calculated risk, there are really no surprises here either. I hope you can see I'm gonna break down this breast cancer risk. My elevated risk report, at the top of it is breast cancer. The average, they say, is 13.5%. That means 13.5 women out of 100 with my Snips get breast cancer. And I have a higher risk with 17%. This is based on 8 Snips, not the BRCA, the famous high-risk SNP. That is in a different report, and that is something I do not carry. So this is on unrelated terms, another kind of breast cancer result, an elevated risk. I have 1.25 times the average risk. And when you look at average risk, it's really not my largest average risk. My largest increase over average risk. Exfoliation glaucoma is, but even when you increase my risk almost 3 times, I still only have a 3% chance of getting it. So this is important when you look at these sorts of reports to consider. And how much more am I concerned about breast cancer now that my risk is 17% rather than 13.5%? And another thing that's trippy about probability is I'm just as likely to be one of the 83 people out of 100 that doesn't get breast cancer as I am to be one of the 17 people out of 100 that does. So I don't know how I'm supposed to think about that, but think about that for a while. My decreased risk, these are all the conditions in their report that I have decreased relative to average risk for. And I still have a higher enough risk at coronary heart disease to be the same as my elevated risk of breast cancer. So I have rotten genes for breast cancer, I have great genes for coronary heart disease, and it's all a wash. Woo! And look at all the latent diseases I have. These are my decreased risks. By the way, these are all the stellar reports that they put their trust behind, and for some reason they show you all of the things that are still up and coming that they don't put their seal of approval on, but that maybe once enough peer-reviewed research is behind that they'll update the report and give it the star treatment. So that's sort of how this works. And then of course typical risk. My typical risk for obesity and type 2 diabetes just by being alive is so much greater than my elevated risk for breast cancer or my decreased risk for coronary heart disease. So that's something to consider as well when you look at these reports. But I think it should be obvious from the rest of the talk that I really don't have my students or me look at these reports for the health benefits of it, for the health information. You can also get your drug response report. And look, I have a I have an increased response to warfarin, which might come in handy to know if I ever needed to be treated for a heart problem in the emergency room. You can look at your carrier status for very simple or relatively simple diseases that are controlled by one or very, very few Snips or alleles, sorry. And this is really only maybe important in an evolutionary context, and that context would be like my mate, my husband. And so when I look at my husband's 23andMe report, if he carried the same alleles for these diseases that I do, then we would have an increased risk of having a baby that would have the disease, that would have a copy from him and a copy from me, and then would have the disease like cystic fibrosis. But when I look at my husband's report, he doesn't carry any of these actually. So we don't have an increased risk that we know of for having a child with any of these conditions, but if he has a de novo mutation for any of those things, that would be a new unknown variant of the allele that causes the disease, but that's just not documented yet, then it could still be possible. And that's why I think a lot of people go to sequencing for these things, just to make sure to find out that they don't have a new unknown or rare type thing. Oh, and speaking of baby making, you can plug in anybody you're sharing genomes with into this inheritance calculator and look at what your babies will be like. And I don't want to think about my baby drinking alcohol, but it won't be flushing in the cheek. It won't be flushing red in the cheeks when it does drink alcohol. It will be tasting bitter. It will have waxy earwax, because that's what our families has. It could have any eye color, which is exciting. It will probably be lactose tolerant, although another interesting finding, my husband and I are the same for this genotype, and he is not good with the lactose. So one hypothesis is that he's got a different microbiome than I do. I've got much more helpful ones that love the milk, and he's got ones that don't so much love it. Of course, the ancestry results are terrific, and as you probably would guess, I have mostly recent European ancestry. In fact, I have entirely 100% according to their analysis. And if we all go far enough back, I need to get all this out. If we all go far enough back, we come from Africa, right? But since then, evolution's been acting differently on the dispersed human populations of the world, and that's left regional and sometimes local signatures in our genomes that have carried forward onto today. And of course, the process continues, but those old traces since that dispersal from the earliest humans occurred, that is detectable in our genomes. So that's why it only goes back as far as Europe, even though I should go back as far as Africa. And then this is another way to look at it. There are tons of ways to look at these data. I'm only going to show you these 2 ways to look at these data. And you can see where I share the most similarity. And it's really with Swedes and Finns and Norwegians and Irish and the folks in the UK. And you can learn how much Neanderthal you are. And if you are more Neanderthal than me, I hope you come talk to me after this is over, if you know how much you are. I am, among my friends, ranked 6th. I am in the, well, let's see. I have 2.9%, which actually isn't that much. I was hoping for more. But it's a pretty good chunk of cave woman, right? And this is what you'd predict. People who have most of their ancestry from Europe or Asia, you predict they'll have at least a little bit of Neanderthal genes, and people who have most of their ancestry from Africa, you predict they'll have very little to none. And I guess that's because, whatever caused this happened too recently for much Neanderthal DNA to flow back into Africa. And I'm sharing genomes with 50 or so people, which is really one of the best things you can do on 23andMe, and see how similar you are to your students, which is unnerving. They're all blacked out here so that I wouldn't reveal them. But what was the best part of this, The best part of this was when I invited my mother to share with me, and I thought, okay, she's got to be at the top of the heap. And then she was. And then I thought, oh no, I'm only supposed to be half of my mom and half of my dad. So if I'm 85% of my mom, does that mean... Yes, it means that a lot of the Snips I got from my mom, my dad had the same Snip. And I know they're not that closely related, but we are all inbred this much. That I could be 85% is the same as my mom is telling you how inbred we all are, that I could get the same sorts of alleles from my mom as my dad. And this is where, have patience with me. I'm going to kind of transcend for a second. I'm thinking, wow, okay, if I'm this close to my mom and to everyone else, I'm really not genetically different from anyone, right? This is what you're thinking, or at least if you're me, as you're getting all these results back and you're sharing your genome with people and you're figuring this all out. And you're thinking, isn't it amazing how much of my biology is probabilistic and influenced by many genes? A lot of those disease reports, for example, were based on many genes. And I know that the environment is a huge influence as well. So it makes a lot of sense, if we weren't so complex, all our little variations wouldn't have wiggle room and all the workarounds to function well. And, I'm just in awe of all of this as I'm accepting this, how similar I am to everyone and how complex it is and mysterious it all is. And then I'm scrolling through my raw data file. You can download this text file with all of your data. And it is huge. And you're scrolling through that raw data file with all those thousands upon thousands of A's, T's, C's, and G's just rolling past your eyes. And you're swept away in the awe that humankind has created the tools and the methods to figure this out. I mean, you've read about it. But when it's you and you're seeing yours, all of a sudden it's magnificent. And not because you're seeing yours, the view out from it to see what other people have done is magnificent. And beyond, biology sure is elegant, but what people have figured out is really what brings somebody like me to tears. But then there are issues. You hear the steady drumbeat of the critics, and they make some fine points, and there are important considerations to be considered. For starters, peering into your genome doesn't just give you your genes, it shows you what your parents and your siblings and your children's genomes look like too. And so what if they don't want to know any of these things that now you know? Because you know theirs. Knowing yours is not just yours, it's other people's. It's also an opportunity, depending on your traits and your sleuthing skills, to detect that your biological father is not who you think he is. And you could also be surprised by your ancestry report to learn something about your ancestry that you never knew, or that changes the way you think about yourself or is somehow disturbing to you. And you could learn something about your gene-based disease risk that causes concern that you'd rather never have known or go without. And so it's not a decision to take lightly to participate in something like 23andMe. And we've been warned to be very wary of companies like 23andMe who are out to make a profit. And although they are tediously open with the databases and they thoroughly cite all the peer-reviewed research that they rely upon to build their reports, some people say that's not enough. And a big part of 23andMe is a voluntary participation by their participants to engage in research that they are doing the way that universities and hospitals do, that they are trying to do to link genotypes to phenotypes. And some critics worry that they're doing this for this for-profit enterprise for motivations that are worthy of concern, but even scientists in universities and hospitals have some very selfish motivations, and some of that includes financial gains, and it's not really clear to a lot of us why a for-profit endeavor is necessarily automatically sketchier than any other scientific endeavor. But then I answer myself a little bit here, and this is where it gets a little or a lot offensive to a lot of critics. 23andMe gives the impression that it's good for your health to know your genome. And they even, in a lot of their PR, they call customers patients. But it's unclear how that could be so, except vaguely, or as supporters might say, except that genotyping, sequencing technologies are just a tad bit ahead of the science, and that we'll be there someday, and that we'll understand these things better someday. That one day knowing your genome will pay off, and that it'll be regularly applicable in a clinical setting. Still, for now, geneticist Ricky Lewis, she's authored many genetics textbooks, says she'll wait to see her genome until our understanding improves, and gene-based disease risk is more than, quote, reading a novel and considering each word in a vacuum compared to understanding the unfolding story. So much of, what am I saying? Okay, so this novel it's a novel of data that really dwarfs War and Peace. And it just seems to be destined to overwhelm doctors with information, much of which is probabilistic or meaningless. And many of these diseases, like type 2 diabetes, are caused by genes and environment. And they don't know anything about your environment when they give you these reports like I just showed you. They don't know anything about you but what your spit said. So my real risk is, whatever that is, could be higher or lower than they report on 23andMe, and I don't know how that really helps a patient. But much of science, okay, here's the other side of the story, much of science, like finding treatments for diseases, is kick-started first by raw exploration. And it's the surest way to precipitate discovery, okay, and lots of it, okay. But then there's the nagging question, what does one do with a risk estimate? On behalf of both the consumer and their healthcare provider's perspective, some have complained that most of the risk estimates from 23andMe are not actionable or could lead to the wrong actions, and that they could lead to a lot of worrying over nothing. And how does one know if they are going to worry, if they're the worrying type, until they are given something to worry about? So, it's also expensive. So it's not really, something that, it costs a couple $100 still. It's still a lot cheaper than getting your whole genome sequenced, which is $1,000, but it's not open to all. And the cost keeps coming down as technology improves. And something that my students pointed out I thought was really a great point. They think that if the high cost is a good thing, and that it could come down to so low that it could be a bad thing. That while it's still a high cost, they think consumers are going to really learn and really read and really try to deeply understand the meaning of these reports. That they're not just going to take the results and run if they invested that much money in the process. But there are many who have far less hope in humanity than that. And not only are you gambling with your money that you'll actually be satisfied with your results, but you're also spending money on what are mostly probabilities and estimated ones. And they could change, and they probably will. And what won't is your genotype, probably. So at least you'll have that because you can download those raw data. But not only are you gambling with your probabilities, but many of those reports are based on a limited data set from a geographically limited sample. So when genetic science is dominated by white people, and it's funded by white people to learn how to cure diseases that affect white people, guess where many of the reference samples come from? And guess then where, guess what a report looks like for someone of mostly Asian ancestry, where there are fewer reference samples from Asia. It's not as complete, and they spent the same amount of money on a 23andMe spit kit. Here's a good example. After I took a survey, they give you the results back of what the participants say, and I'll zoom in on it. It says, are you lactose intolerant? And for 23andMe participants, most people said no. And that is the opposite for our species. Most people in our species are lactose intolerant, so it's not really representative of what the data they're collecting and what they're working with to link genotype to phenotype isn't really representative of everyone. And that gives you a good idea of who's participating in 23andMe as well. And of course, the worst implication of all of this is that it will reinforce notions, either out of ignorance or stubbornness, that everything about you is programmed from birth by your genes and can be predicted by your genes, personality and all. And political correctness doesn't matter here, because regardless of which side feels better to you, the evidence is plain as day. Genetics doesn't work that way. But here's the catch. Yes, everything about you is genetic. At some level, it is, or else it's magic. And to resist the power of genes is fairly common in our culture. But understanding that everything involves genes is not to be genetically deterministic. And your memory is a really great example. You can't make memories without your genes, but your genes don't contain your memories, especially not your future memories. And here's another good example. It's recently published in PNAS. It's a nice new demonstration of how three-dimensional vision requires stimulation to develop. This thing that many of us rely on our entire lives is not pre-programmed by our genes. Yes, genes are involved in developing this, but stimulation is required. This was a nice little study that showed that regardless of how early or how late you were born, you still developed 3D vision at the same pace. So it wasn't something that was timed genetically. It was based on stimulation from the environment. So environment and experience through time and space, these things affect your biology during life and in different ways, even if you are an identical twin. And further, there is so much complexity in our genomes. Most of the variation we see is not discrete, on or off, this or that. And it falls along a spectrum, like height, like weight, like body fat percentage, like skin pigmentation. And this sort of variation can be caused by and influenced by many different phenomena. For example, a single gene can be involved in many different processes in your body. Also, one gene, depending on the context, either in your particular genome, in your particular organ, in your particular body, in your particular city, in your particular planet, can lead to various different biological outcomes. We are creatures who are integrated into our environments, totally plugged in, not hermetically sealed like astronauts or scuba divers, and we're built for it, and we require it. And all these lessons about determinism and complexity are either lost on critics who are ignorant about biology, or critics of personal genomics believe these lessons will be lost on us ignorant consumers. But there are also privacy issues. Madonna apparently has all of her DNA trail wiped clean wherever she goes. This is mean on camera laughing at Madonna. This can't be safe. In principle, anyone with sufficient knowledge could take a person's DNA and use it to infer paternity or other genealogical information to claim statistical evidence that could affect employment or insurance, claim relatedness to infamous criminals, plant incriminating synthetic DNA at a crime scene, intentionally or incidentally reveal susceptibility to diseases currently lacking a cure, reveal maybe that you have a Y chromosome and people think that you're a female, things like this. And all of these without getting your consent. So that makes people wonder, how secure is your genome with 23andMe? How secure is your genome where you're sitting right now? These are good questions. But why do we care how secure it is? Are any of these real threats? Are any of these real threats? A common worry is that the government in the future could use our genomes against us in any number of ways. But I can't help but think, like in a world where our government is using our genome against us, that is not our biggest problem. Okay, and then there's the concern that personal genomics and related technology are encouraging a new wave of eugenics, with fertility treatments enabling people to reproduce without passing along dangerous mutations. That's called positive eugenics, or good eugenics, with products that allow parents to choose the sex of their baby, that allow mates to choose mates based on genotypes. I heard maybe dating sites are going to start putting genetic information on them, or maybe they already have. I'm beyond that. I'm married. But you can't help but wonder, now this is something I've been thinking about for a while. You can't help but wonder, how different is any of this from what we've already been doing? And who's to arbitrarily put their foot down and say, right now is when our playing God has finally crossed the line and we've got to stop? Because information has long influenced human evolution. The discovery of the birds and the bees, I'm talking about figuring out that intercourse is what makes babies, meant that people could choose who they make babies with, at least a little better than before when they didn't know that's how babies were made. And meant they could influence who others could make babies with. And our unique reproductive consciousness has built a tradition of intentionally building babies with certain favorable traits. And it runs deep in human history, long before the DNA molecule was ever discovered and 23andMe was giving people spit kits. And if this is my fun little joke, if this is hard to swallow, think about puppies, think about ice cream, think about bacon. All of those things are thanks to this information as well, this reproductive consciousness. And so this is information that's been affecting evolution of ourselves, evolution of all these plants and animals around us for as long as we've had this information, and much longer, much deeper in time before we knew anything about DNA. There are fears with increasing genetic knowledge that will support racist assumptions of vast differences in valued traits between continental groups. But that obviously doesn't take DNA either. It has not taken DNA in the past. And there are concerns about what geneticist Richard Lewontin recently wrote and recently described as, quote, the concept of an authentic self that lies hidden and unexpressed, but which in some sense is the essence of what I am, even if unperceived and without any basis in scientific demonstration. The concept of a self that is an authentic essence but not clearly perceived suggests that my manifest properties and attitudes are a mere patina, and that in ways I do not recognize, my inherited inner self, my DNA, is struggling to assert itself. So he was criticizing this approach to genetics. And it's a good question. Should we worry about whether personal genomics participants are seeking to dig out their soul? But then you wonder, even if it does seem to indicate a level of biological ignorance, is this any of our business, whether that's what they're looking for or not? So there are these judgments against consumers who are using 23andMe and products like it to sort of like Ouija boards or the Zodiac or their horoscope. But this is what humans do, and if we didn't have DNA for this, we'd be using blood types. And we already do that. So genetics as this crystal ball, it's such a popular impression that it's even enough to keep folks away from personal genomics. And it's seen as a window into your imminent death. And I think few people find that appealing. But 23andMe cannot tell anyone how and when they will die. Not even close. In fact, all the things we already know about life expectancy still hold. If you want to extend your life or lower your mortality, just watch what you eat. Don't smoke. Don't drink. Don't be polluted on. Don't drive. Don't have children. I didn't say it. It's science. And don't have a Y chromosome. You'll live longer without it. So right now we've got, we've got all these people, these professionals, geneticists, doctors, theologians, ethicists, historians, futurists, capitalists, all over the spectrum about personal genomics, about the public having access to their own genetic information, even if we don't understand very well what that information means. So just to end this off, I want you to play along with me for a second. I want to try to make an analogy. I want to know what you think is going on here. Don't say it if you've seen this before. Many of you might have seen this before. But if you haven't seen this before, shout out what you think is going on here. Dancing. Okay, I heard dancing. Hugging. Okay, anything else? Getting eaten. More. Petting a dog. It is a dog. It's an Irish wolfhound. And it's Grover Krantz. He is now an infamous anthropologist whose remains are on display in the Smithsonian with his beloved Irish wolfhound Clyde, as he wished them to be displayed. And yes, it's very much a loving embrace and not being eaten to death. But that's the other way to see this. And that's why I think, you can see love and life, or you can see pain and death. And I'm not trying to be too simplistic about this, but I think this is a nice analogy for the different perspectives on personal genomics, the different ways that people perceive them. So I obviously fall along this end of the spectrum. And I'm one of the many out there who wants to have all the information, even if it's not ready for prime time yet, even if interpreting much of it requires fancy brains and fancy computers. And I'm optimistic about it, but I also know very well that there's so much about our health, our lives, that we'll never be able to predict. Even though we have scientific explanations for those things, we won't be able to predict the future about those things. And I'm comfortable with that, with how science works like that. And I also don't see the health aspects as a significant result of this experience. I also don't see my genome as part of my identity. Or maybe I do, and either way, it's not a problem. And like me, although he's a lot less exuberant and a lot more knowledgeable about genomics, Misha Angrist, a friend of mine who's a participant in the Personal Genomes Project, where he has his genome publicly sequenced and publicly posted online, and he's also the author of the must-read book, Here is a Human Being. He writes that on seeing his full genome for the first time, he writes, quote, "We were ready to find out for ourselves," the personal genome participants, "not because we have any illusions about genes as the secrets of our being, but because we don't." But we should be sensitive to those who have a less than thrilled outlook on personal genomics. And I think this awareness is important for each of us as much as we all march into this genomic era. And there are myriad reasons to have this, to live on this other end of the spectrum. And I do, however, think that a lot of the folks who are there would move closer to the middle if they understood genetics better. And what better way to understand genetics than to hold your nose and plunge into personal genomics? But that's the pickle, right? Like to be informed, to really learn about how this all works, you've got to be a participant in it. The thing that you do not want to do, the thing that you fear. But out of all my 130 anthropology students, I only got feedback from one who said he or she had a negative experience out of all 130. And what I'm witnessing as a result of this new curriculum is the most engaged classroom I've ever seen, the most contemplative, the most invested, the most willing to understand details as well as larger, more complex issues and phenomena. The genie is out of the bottle. Personal genomics are here. They're being marketed to us. It's imperative that we understand genetics, their powers, their weaknesses, their negative and positive potential. And as Skip Gates of Finding Your Roots and the Harvard University professor said to Misha in his book, quote, "You can't be a Luddite You can't stop it. You have to try to understand it. We have to become scientifically literate so that we can learn how to intelligently challenge the potential abuses. And what I'm finding with my students is that their learning has extended beyond to evolutionary thinking. Beyond genes, beyond anatomy, physiology, disease, culture, it's really extended to evolutionary thinking. Seeing your genotypes improves your understanding of evolution. And personal genomics shows us that genetics can, in fact, work the way that scientists say it does, the way we've only read about until now. It shows us that scientists are pretty good at discovering and explaining, and so maybe it's not one big conspiracy, this whole science thing. Maybe global warming and evolution are real after all. It shows us that we can be skeptical of scientific methods and claims without being anti-science, and that predictability is not a requirement for scientific explanations. Personal genomics shows us that we're not so different from everyone else. It shows us that our Snips can be traced back tens of thousands of years. My maternal haplogroup, for example, 60,000 years ago in the Middle East, the U group I belong to. But that's just one lady back in 60,000 years ago. One lady, but still. There's a real person back there, and personal genomics gave me that person. It shows us that our Snips can be traced back that far. It shows us that not just because some anthropologist said so, but because they really can. It really is true. It shows us that variation is the norm in biology and that complexity is the norm too. It's normal to be different. And it shows us how those things can come about through so many different processes at the individual, population, and species level. It shows us that what we find fascinating about people, other people, and ourselves is not necessarily tied up in genetics. And when it is shown to be genetic, it's harder to justify value judgments we make about that variation. Another quote from Misha Angrist from his book: "Thinking DNA is cool. Thinking DNA is cool, even in the face of its often limited predictive value, hardly makes one a genetic determinist. Yes, we are fixated on genes in part because we find them to be seductive and reductive explanations for human traits and behavior, but we are also fixated upon them because, like the proverbial drunk looking for his keys under the lamppost, we can be." So most of us will never explore outer space. With personal genomics, though, we have a new frontier to explore. We have inner space. And we don't have to do anything but spit in a tube and connect to the internet. It's almost magical. It's a brand new view into the past, the present, and the future, and your place in the species, your place in the ecosystem, your place in evolutionary history. This is one really heavy tool for triggering like Emersonian transcendence. So you know the Earth is round and blue, but what would it feel like to see the color and curvature for yourself from space? You know that Mars is there and what it looks like, but what's it like to walk on it? You know there are rare fossils of your ancestors from millions of years ago, but what does it feel like to hold those in your hands? This is one of those moments, and it's down here on Earth, and it's not locked up in a museum vault halfway around on the other side of it. It's right inside each of us. Each of us is a window into humanity, its shared origins and its ancestry, all the way back to the first breath of life on Earth, and even deeper. To know thyself is to truly know the epic that is humankind. Thank you, California Academy of Sciences, very much for hosting me tonight. And thank you to the Leakey Foundation for so much support and for making this a thrill for me.